All right, so I wanted to use this video to introduce the extra credit and give you like a little example because you can read just fine, but turning what you read into what you're doing, that is an art. So here we go. So this is the instructions for the extra credit. And so let's go over them real briefly. So at the top, it's just saying that virology, yeah, some groups get studied better than others, like some groups of viruses, some specific viruses. And but we're going to be looking at RNA capping because everything that's a virus has to express some sort of gene, and the only way to express a gene is to somehow generate a messenger RNA. Now, whether you do that with host enzymes or whether with viral enzymes, yeah, it remains to be seen. And we're focusing even more. We don't care how the RNA itself is made. We're caring about how it gets capped because that is a problem. It is easier to make a string of RNA than it is to cap it because you can make it basically with one enzyme and a primer of some sort. But to cap it, usually it takes 4 enzymes. Sometimes you'll find a virus that'll double up. It'll use one enzyme to do 2 functions. But quite often you will see viruses that have 4 separate enzymes to do the 4 separate functions, which is really cool. All right, so here we go. And so we're going to look at the level of a virus family. And so down below, it's the same virus families. You remember these guys from the homework, so like Coronaviridae, awesome. And the nice thing is that you can use any virus from the Coronaviridae to complete any part of this. Like somebody may have figured out the entire capping system for feline coronavirus, and that's fine. You can just use that. But if they did one enzyme from feline coronavirus and they figured out another enzyme from SARS coronavirus 1, that's still okay. You can put those together, and we'll assume that if it works for one member of the family, it's more or less the same in all the other family members. You don't have to document like the guanylyl transferase for every single virus in the family. That way lies madness. Yeah. All right. So here's what you're looking for. There are 4 enzyme activities involved in making a cap. They're taking off of phosphates. We call that terminal phosphatase or gamma phosphatase from RNA. There is transferring a guanine, so it's a guanine transferase, guanylyl transferase, something like that. There's putting the first methyl group on, which is an N7 methyl transferase, or there's putting the second methyl group on, which is a 2'O methyl transferase. And so what we're going to do to document these is you're going to go out and it's like a giant Easter egg hunt. You're going to see which ones you can find in all of virology literature. And so, all right, how do you do this? Well, what I want is I want to know what paper tells us this and what specific figure. So here, I want to find out where's the experiment on which this knowledge is based. So there are papers that we would call reviews or introduction sections that'll say, oh yeah, it has a guanylyl transferase. Yeah, and they may cite a reference or not. If they cite a reference, that's good. That's usually another paper that may actually be the paper that tells you where this is. So that's one way to find them. The other way to find them is just through Google Scholar, and I'll show you an example of that in a moment. But we want to find the paper, and then in the paper, I don't just want you to find the words like, oh yeah, it's totally a guanylyl transferase. I want you to find me the figure where they show it. And they're going to show it with some sort of laboratory test. A lot of times it's going to involve radioactivity because these may be old papers, and that's how science was done back in the nuclear age, like the 80s. Yeah. Yeah, and we wanted to see if we can document each one of these. You may get to a family where you can't document any of these. And that's okay. That's just a gap in science. There are no extra credit points we can get there. You may get to a family where all of these things are put on by host cell enzymes, like by a human. If that's the case, you just figure out, okay, how does a human cell, what's the enzyme that does this? What's the paper that showed it? It's fine. Same thing, except that you're talking about, what normally happens in a cell rather than what happens, something that the virus is bringing along. So once you find that paper, then I'm going to tell you how you cite it. So one, give me a fragment of a sentence, like a phrase or something, that tells me what this figure shows that you're pulling out. And so something like, they transferred 32 phosphate, that's like radioactive phosphate, labeled GTP onto the end of an RNA segment. That counts. And that is transferring guanine to RNA. That is guanylyl transferase. That's literally what it means. So there's no better experiment than that. Yeah. Then you're going to cite the paper and the figure that it came from. So you give me that little fragment, and then you're going to cite the paper and figure. You can put that in any format. Google Scholar is great. It'll flip between the formats. If you just call up the paper in there, you can select which one you want, and that's fine. And then you need to tell me which figure it's coming from in there that your sentence is referring to. Yeah. And again, it doesn't have to be a sentence. It can be like a bullet point. It's fine. All right. You'll run into some weirdos, because this is a big problem for viruses, and they've come up with a lot of creative, very virusy solutions. Terribly jury-rigged, just improvised, garbage solutions, like you would expect from a virus. So maybe you have a virus that doesn't have a traditional cap structure. Sometimes they'll have a bit of folded RNA that acts as if it was a cap. Sometimes they'll have a protein up there that does the job somehow of being a cap. Sometimes it's a combination of the two. That's okay. In those cases, you're going to find me the papers where they demonstrate that this piece of RNA or this protein attached to the end of an RNA genome is what actually makes this stick to the ribosome in a way that it gets translated. Yeah. Review articles will be your friend in this, because review articles are by people already in the field, and they can point out, they're supposed to know where these things come from, where these ideas originate. And so you'll hopefully find a couple of good reviews for a family, and that'll get you started on there. Of course, if it's not known for sure, on some of these papers, someone will have done bioinformatics. And that just means it's a way of using the computer to analyze the sequence of the virus and say, oh, you know what? This has real similarities to a guanylyl transferase or an N7-methyltransferase, and so probably that's what it is. If there isn't a direct demonstration of enzyme activity, which there isn't for a lot of these, it's okay. Cite me a paper that uses bioinformatics to correctly predict. You won't know if it's correct, but that uses bioinformatics to predict these functions. All right, now, how's this thing graded? Well, all right, here's the nice thing. 1 extra credit point, basically for each documented thing. So if you can document an enzyme activity, that's fine. There may be multiple ways to do this. Someone may have identified the activity before they figured out which enzyme actually did it. They can say, it's about an 84 kilodalton enzyme. I don't know what it is, but it did this. And later on, they may say, here, we found this enzyme, and it does the same thing as they showed in that other paper. And as long as each paper has an actual demonstration of what the enzyme does, that's good enough for me. In some cases, it may be a structure paper. They may solve the structure of the enzyme with a little cap molecule and a piece of RNA right inside of it, and that is also good enough evidence. So for the more popular viruses, there may be several different papers and figures that you can cite that are all actually just fine, for the purpose of this. So, see what you can do. See what you can find for me. Each one that you get is an extra credit point. Every time you can document a non-traditional cap structure, that's 2 extra credit points. And these are due before exam 2. So that's where we're going to pause this